Hello, everyone, and welcome to another session of the HPP Innovation Week. My name is Rui Queiroz. I am part of the applications team at Hyperbaric, and today we'll focus on the effect of pressure on proteins and other macromolecules. Generally speaking, pressure does not change the nutritional value of foods, not affecting small molecules like vitamins. However, the conformation of big molecules like proteins can be modified by high pressure, which can be a useful tool for product development. Today, to talk about this topic, we have a great speaker, Dr. Vivek Orlian from the University of Copenhagen. She is a food chemist with a PhD from the University of Copenhagen, where she is now an associate professor in the Department of Food Science. Her research area is thermodynamics, with emphasis on high pressure in food processing and its effects on food components and their functional properties. In this session, she will cover the use of HPP for structure modification of food components. Thank you for joining us, Dr. Vivek, and the floor is now yours.
Thank you, and thank you for inviting me and giving me the opportunity to present my work on high pressure. So high pressure, as research, can affect macromolecules, and I will present an overview of some of the studies from others and my own on how to modify meat proteins and milk proteins in relation to functionality, in relation to structuring meat and milk products. As we may know, that meat are constructed of three major proteins types: it's the collagen, the sarcoplasmic proteins, and the myofibrillary proteins. And that is composed into the meat structure. And if we look close to it, then we have the ultrastructure of the myofibrils that are composed of the myosin and the actin molecules, basically. As we can see up on the right corner, we have the thick filament and we have the thin filament. And this is what gives the meat muscles. When we start pressurized on meat, a whole meat muscles, for example, then we will soon, or first we will have the thin filament, so that is the F actin and the Z line, that will start to break up. And then it will release the thin filament and the thick filament kind of from each other. Then if we increase the pressure, we will have a loss of the M line, and the Z line will totally disintegrate. So we will have a I-band rupture, and we will have a depolymerization of the actin protein molecules. At the same time, the myosins that are set into this myofibrillar structure will also be disrupted, and it will release the individual or single myosin molecules. If we keep on pressurizing, increasing the pressure, we will also affect the individual myosin molecules, and it will disintegrate into the heavy chains and the light chains and what is called the head. But depending on, of course, the system, now this is a whole meat, we can also have a minced meat, or we can actually have the protein in solutions. We can either have a total solubilization of the myofibrillar proteins, but we can also see an effect on the insolubilization of the myofibrillar proteins. But what most of the research that have been published have found out that we have this pressure around threshold pressure, around 400 megapascal, where we see a difference in whether the myofibrillar proteins are solubilized or insolubilized. And the reason for the insolubilization of these myofibrillar is that they actually aggregate together again, of course, in another way than the original native myofibrillar structure. And there have been suggested three models for this integration or aggregation of the high myofibrillar proteins. The first one and the early one was by a Japanese group, saying that when we have these individual myosin monomer that had been disintegrated from the whole myofibrillar structure, then they can be associated together again. So we can have, as we can see here, that the heads of the myosin monomer can be set together, or they can be put together in this monomer of 3 or more association of the individual myosin protein molecule. And then that can really result in a aggregate which they were called daisy wheel because it looks like a wheel. Then there was also a suggestion that when we have the individual myosin molecule, we can have a total disintegration of the protein. So we have the tail and the head into kind of small pieces or peptides, and then they will aggregate or join together into a similar daisy wheel, which can then form together more wheels together, which can eventually become a gel. And the last model suggestion was that we have the individual myosin molecule. Then we have a dissociation of the heavy chains, but the head part is still intact. And then it's these heavy chains or tails that will aggregate and form a gel structure. So there are these three models, and most of them are depending on a little whether you investigate the myofibria proteins in a whole meat or a minced meat, or you actually extract the molecules or the proteins before high pressurizing. So the starting point of your investigation also affect what you actually see. We have tried an investigation on a whole piece of meat, which we high pressurized. Then following that, we took some different regions that are known to break specific molecular interaction or bonds in order to investigate specific reason why we see these aggregation. So the first three graphs here we see from the B, C, and E that if we from the B, if we add a phosphate buffer after high pressure of the meat at these different high pressure levels 200, 400, 600, 800, we can see that we do not break electrostatic interaction. And the reason why we can see that is that this is the soluble protein we have from the raw meat. When we pressurize, the hypothesis is that we have these aggregates that are not solubilized. And as we can see, the soluble protein content decreases, meaning that we did not break electrostatic interaction. And the same goes on from the DTT, which should break disulfide bridges, that this is not the reason for increased solubilization. And also thiourea, which will break hydrophobic interaction. We can also see that we do not get an increase in protein solubility when we pressure at 400, 600, or 800 megapascal. Then if we add urea to the pressurized meat, then we can see that at all pressure we have a considerable increase in the level of the protein that we can solubilize afterwards. So the conclusion is that high pressurized meat kind of get tough, or the structure is changing in this specific case due to hydrogen bonds. But as we can see, this is just for an overview. A lot of study has been done on high pressurizing and meat in order to see different properties. It could be water holding capacity, lipid oxidation, the amount of some of these interaction, the protein solubility, and also even if it's the sarcoplasmic protein or the myofibrillar protein. And as we can see, there is a lot of differences, whether it's increasing the amount of, for example, the tile, the reactive tile, or decreases. So a lot of study show different results, and typically it could be depending on both what is the pressure level or what is the meat type. So there is still a lot to investigate, but in generally we can say that the overall mechanism of myosin dissociation and aggregation is that first we of course need to rupture this filamentous structure, so the myofibrillar structure is dissociated and we get these thin and thick filaments, which can then cause solubilization of the individual myofibrillary proteins. So we do have some protein denaturation due to the rupture of these non-covalent interaction. Then when we have the individual proteins, so to speak, some of them are quite reactive, and if we then increase pressure above 400 typical, then they will aggregate together again and make larger aggregates that cannot be solubilized. And that's actually what can be used in designing new structure of meat or meat gels. It's a little the same as when you have a raw meat. When you cook it, it becomes harder. Sometimes it becomes tender when you eat it. But if you cook it too long, it will get hard and very tough to eat. It's the same. It's kind of the same analogic we have here in the high pressure. So talking about that, then we try to actually use high pressure to produce some sausages. When you do sausages nowadays, you have to add a lot of salt because salt will solubilize the proteins. So when you cook the sausage, it will form a gel, and you will have instead of the mincemeat, you will actually have a meat gel. But of course, as we know, there are some health problems with eating too much salt. And at the same time, when you cook or add to salt, you also impact flavor in a way. So we try to high pressurize some some sausage, sorry, to see what happened. And first of all, we can see that when we high pressurize, here we have 3 different pressure levels. We have 400, 600, and 800. And I can see that I have mixed around. So it is actually the white that is 800. But anyway, when we increase pressure, we increase the hardness. This is a measure of how hard the gel becomes. And we can also see that we can see it for more of the samples. So even here we have a high salt value on 1.8. Here we have a lower salt value, 1.2, and we can see that the texture hardness is almost the same when we have pressurized the sausage, meaning that we actually don't need to have that high salt level. We can also see that if we increase temperature during the pressure treatment, that will also affect the hardness of the sausage. So when we have high temperature and high pressure, they will act kind of synergistically, or at least the effect will be larger, and we will have a harder sausage than if we only used pressure at cold temperature. And that can typically be due to that, as we know, temperature also facilitate protein denaturation and protein aggregation, which we know from when we fry egg whites. We need a certain temperature because before that will aggregate and make this firm texture. And then we can also see that if we add, for example, potato starch, and we also have some carrot fiber, we will also get an increase in texture. So we also have some additional hardness of the gel when we add some starch or fiber. So adding some healthy components instead of salt could also make a harder sausage. So at the end, we can use high pressure to structure meat-based products. It will depend on the structure, of course, and the functionality of the myofibrillary proteins. So here it's the pressure level that is the key parameter. We need to be above 400 megapascal because we need to have the proteins solubilized and denatured in order to form the molecular network that gives the gel structure. We can then play around with the texture sorry, by combining with heat treatment or incorporate other kind of non-meat ingredients. and in that way you can actually also make the sausage healthier with adding fiber, for example. Then milk is also a kind of very complex example of different proteins. In milk we have the beta-lactoglobulin and we have the caseins as the proteins that provide most when we do some milk structuring or working with milk texture. So if we start with the beta-lactoglobulin, we have A here, that is a native beta-lactoglobulin. It's a dimer. So we start pressurizing this one. We will kind of hydrate each of the individual protein molecules, because as this dots is a representative of the water shell we have around the molecule. So when we start pressure, we kind of pressure the water into the three-dimensional structure of the protein, and then they will dissociate. And if we increases the pressure even more, this water will also be pressed in, kind of, you can say, into this structure of the individual molecules. And then we will have a protein molecule that is called a molten globule protein. And the reason is that this has not a specific structure as a normal native protein will have, but we have this kind of soft structure of the molecule of the protein, and that could have, and this structure can be in many different ways. But if we then keep on pressurizing, then it will eventually totally denature. So this kind of molecular or round structure will open up, and we will have this a little longer or spread out protein. And when we have that, then we have all kind of interaction that can also aggregate the beta-lactoglobulin together, and that will also be either aggregates or a gel. As we can see here, the threshold for the protein denaturation for beta-lactoglobulin is 300 megapascal. So this is kind of the level where you can say either nothing severe happens or something really affects the protein molecules. And milk also composed of the caseins, and they are held together in the so-called casein micelles. So we have a lot of different caseins. We have 4 different types of caseins, but we also have something called colloidal calcium phosphate that kind of keeping all these proteins together in this round, not molecules, but micelles. This is some of the reasons why milk are white. When we start pressurizing on these micelles, water will also be kind of pressed in the micelles, and we will disrupt whatever there is of electrostatic or hydrophobic interaction, and it will start to dissolve the micelle. So above 200 megapascal, we will have some individual caseins. These are the long one here, and we will have some calcium that will be released from the calcium phosphate, and we will have some minor micelles also. And then if we keep on pressurizing, depending on the pressure level and the time we actually have the pressure holding, we can have different outcomes. So if we increase pressure above 300, we will further dissociate the micelles or have individual casein released. But if we keep this partly dissociated micelles between 200 and 300 megapascal for a longer time, so above 30 minutes or something like that, we see that some of these smaller submicelles will actually join together again, associate together, and form a little larger micelles. But whatever we have this situation or the high pressure where we have a lot of dissociated micelles, when we release pressure, we will have this structure where we have smaller submicelles and some individual or released casein. And these submicelles can be as large as the original or native micelle, but it will not have the same structure because they have been torn apart, so to speak, but they found together, but in another way and structure. The threshold pressure is actually also around 300 megapascal for going from the from the native micelle into these submicelles. And these submicelles and the individual caseins together with the beta-lactoglin can be rather reactive, which we can use to form milk products. But first we just have this one because it's interesting for the micelles dissociation is that we try to investigate what is the impact on temperature or pH of the milk before we pressurize the milk sample. And we can actually see that the starting point is quite important for how pressure will affect the outcome. If we change temperature while we pressurize the milk, we can actually see that we don't get the same dissociation, or we get the same dissociation, but we don't get a more dissociation or less dissociation. So the casein micelle dissociation is more or less independent on the temperature. On the other hand, if we change the pH of the milk before pressurizing, we have a huge effect on the dissociation. This shows what we call the threshold pressure, meaning that with something called high pressure spectrophotometer, we could actually see or measure when do we go from the native micelle to these dissociated micelles. And we could see that changing pH has a huge effect on at what pressure level this will happen. And we can see at the higher pH, we need a higher pressure to actually facilitate this dissociation. So the micelle gets kind of stronger when we increase the pH, and thereby is kind of harder to dissociate. Then we tried to make some milk gels. We simply took some whole milk, or we took some, first of all, we took some skimmed milk whey protein isolate and added water in it. And then we try to pressurize, and we can see if we have water and 50% of whey protein isolates, we can make a gel. But we can also see that if we take milk and add whey protein isolates, we can also make gels. This is a measure of how hard this gel gets. And we can see we have some soft gels, and we have some intermediate gels, and then we have some really strong gels. So we have these three kind of different gels. The strong gels is gels like we can see on the photo here. So they are self-supporting and really strong. And for for some of those gels that were really strong, they were more like a rubber ball or something like that. So for making a milk gel, we can see that it's quite pressure and concentration depending. And as I said, it's also depending on what solvent we use. So if we use water, we get a intermediate gels. But if we use milk as a solvent for the whey protein isolate, we get some really strong gels. And this is because in milk we have the casein micelles. And as we just saw, if we increase the pressure, then we will dissociate the micelles. We will have some reactive casein, and we will have some submicelles that also will go into this beta-lactoglobulin gel network that we have made based on the whey protein isolate. So for summary, it is actually possible to use high pressure to change functionality of both meat and milk proteins. And in that way, we can tune our gel structure or gel molecule network and get different kind of texture. For meat, the threshold pressure is around 400 megapascal, where we can say, okay, we need to be more or less above that in order to play with the texture, depending on the system and the meat type. But we can do some gel meat products where we can actually reduce the salt, and we can probably also have another taste in order compared to normal sausages. The whole point in using high pressure for making sausage, for example, is not just to reduce salt. It also makes a considerable different texture when you eat it. And then actually, because we know that high pressure also can kill bacteria, we can actually have kind of a cold sausage. So we will also have maybe an improvement of the sensory assessment, the taste, because we don't destroy something with the heat treatment, for example. We can also make milk, new milk product, new milk gels. So if we increase the pressure above 300, we will denature beta-lactoglobulin and we will affect the micelle. They will dissociate, and we can make these milk gels. So we can actually produce a neutral yogurt. We all know the texture of a yogurt. Yogurt are made by acidification. That's why it's sour. But we can actually make a similar texture and even a harder texture with high pressure, but without adding acid. So we don't have this sour taste, but have a completely new taste and structure, actually also, and mouth feeling. And of course, the HP yogurt or gel will also depend on the milk cell system and the processing procedure. We could see when we play around with the pH of the milk, we'll have different gels. So if you think that high pressure was very interesting, I will just make an announcement and hope that you will join our high pressure conferences. So we have soon a high pressure conference in bioscience and biotechnology. Here we will have the themes about food, general about micromolecules, biotechnology, and cell biology, and there will also be something about marine science and microbiology. And it involve all kind of subspecialties within this. So it's a broad one, but it's quite interesting, and you can just see the keynote speaker here. And when you're interested, you can go into this website where you can see the program and all the details and information. You can also follow us on LinkedIn. I have to say that the deadline for registration is the 20th of June, but we will also have an online version that will be announced on the website. But anyway, feel free to mail me if you have any questions. So thank you for the intentions, and thank you for having me making this presentation.
Thank you for the great presentation. It was very interesting. If you don't mind, we have a couple of questions for you.
Yes.
The first one is, you focus your presentation on animal proteins, but plant proteins are very popular these days. Can or HPP also improve their properties or functionality? And if yes, how can you use it?
Yes, I will also expect we have started looking into high pressure on plant protein also, and I've been reading a little about it. So it will also affect the plant protein, but in a different way, because of course plant proteins have another structure than meat and milk proteins. They are more easily affected by pressure. But we will also have, because it's the same interaction that holds the plant protein together in their tridimensional structure, that do it for the animal proteins, and so we will affect these one also. The plant proteins are held together in a little stronger way and will dissociate probably at a higher pressure in another way. But I think we will affect those and maybe solubilize them. I'm not sure whether we will have them aggregate in the same way, but it also depends on, for plant proteins, it depends much more on what the situation is. Is it a whole plant system? Is it a plant or a bean? Or do you have a protein extraction and a solution of protein? If you have a solution of plant protein, that means that you have already extracted them from the plant or the legumes or cereal, whatever it is, then we can see that they will also aggregate and make some kind of gel. They are a little, what can you say, more difficult to guide right now, and also they are not so fond of holding the water. But it is possible to make a plant protein gel. It could, for example, be a tofu kind of stuff. We have tried that.
Okay, very interesting. Let's see if we have some more research in the meanwhile.
Yeah.
And the other question is, most companies nowadays use, or all of them use HPP to increase the shelf life or safety of their products. Why do you think there aren't many companies exploring the potential of pressure to improve the properties of proteins and develop new products? Do you have any idea?
Yeah, I think that first of all, it's because it's kind of started with the shelf life, and you can kill bacteria, and you can do it in a short time. So that's the first point. When you have to use high pressure to modulate the protein structure, we need to pressurize for maybe 5 or 10 or 15, maybe even 30 minutes. So that's quite long processing time. And the capacity of the machines has not been that big. So the production facilities or the production amount is less. And the other thing is that I think that it has not just come around for the company yet that is actually possible to structure the protein and make these. I would say it will probably, to start with, be more specialized products, and when they are specialized, they are also more expensive. I remember in the beginning, the high pressurized product for stabilizing shelf life was also a little more expensive, and it was a superior product. So in that way, I think that they more or less just need to see what is going on and get started. And I think that eventually we will also see high pressure used for modulating structure, because we can get new structure. As I just showed, new texture, new gel types, for example, in the milk, and can also be in the meat, where you can reduce salt, which is used a lot in meat, also for shelf life, but also for taste and functionality. And I think that then the third challenge, or why it's not come so far now, is that we are still learning to actually do it right every time. There are still some, as we can see, there are a lot of different outcome of it. We still need to prove that we can get exactly the same texture every time, so we can control what we are doing.
Okay. Now let's see if with more awareness and research we can start finding products with
I
new
hope so.
textures, and yeah, we also. Um, Thank you, Dr. Vivek, and with that we conclude our session. And thank you all for attending this event, and we will see you in the next one. Bye.
Bye. Thank you.